Today we will learn about immune response. Here we will study about humoral immune response. Primary under this we will study about primary and secondary humoral responses, fate of antigen in tissues, production of antibodies, theories of antibody formation, monoclonal antibodies, factors influencing antibody formation, and then we will study about cell mediated immune responses. Here we will study about induction of CMI, cytokines, detection of CMI, transfer factor, and then we will study about immunological tolerance. So let's begin. Let's begin with introduction. The specific reactivity induced in a host following an antigen stimulus is known as the immune response. It is of two types. First is humoral or antibody mediated immunity. Second is cell mediated immunity. Antibody mediated immunity, in short AMI, provides primary defense against most extracellular bacteria and helps in defense against viruses those infect through respiratory or intestinal tracts. AMI also participates, that means type 1, type 2, type 3, hypersensitivity and certain autoimmune diseases. Cell mediated immunity, in short CMI, protects against fungi, viruses and intracellular bacteria like M. tuberculosis, M. leprae and parasites such as Leishmania and Trypanosoma. It plays an important role in allograft rejection, graft versus host reaction GVH, and mediates the pathogenesis of delayed type 4 hypersensitivity and certain autoimmune diseases. It also provides immunological surveillance and immunity against cancer. Both AMI and CMI usually develop together through at times one or other may be predominant type. Now let's know about humoral immune response. Antibody production follows a characteristic pattern which consists of various types. First is lag phase, a log phase, a plateau, the phase of decline. So let's begin with the lag phase. The immediate stage following antigenic stimulation when no antibody is detectable in circulation. It is a lag phase. Then log phase. There is steady rise in titer of antibodies. A plateau. Third is a plateau. There is an equilibrium between antibody synthesis and catabolism. Fourth is the phase of decline. Catabolism exceeds the production and titer falls. Now let's know about primary and secondary humoral response. The primary humoral response has a long lag phase and low titer of antibodies that do not persist for long. In contrast, the secondary humoral response has a short or negligible lag phase and a much higher level of antibodies that last for a long period. The antibody formed in the primary humoral response is predominantly IgM and in secondary humoral response IgG. The duration of the lag phase depends upon the nature of antigen, its amount, route of administration and species of animal. With some antigens such as diphtheria toxoid, the lag phase is primary humoral response may be 2 to 3 weeks, while with pneumococcal polysaccharide it may be as short as a few hours. A single injection of an antigen helps more in priming the immunocompetent cells to produce the particular antibody rather than in actual elaboration of high levels of antibody. Effective levels of antibody are usually induced by subsequent exposure to the same antigen. Due to this reason, the killed vaccines are given in multiple doses for immunization. The first injection is known as priming dose and the rest as booster doses. A single dose of live vaccines is sufficient as multiplication of the organism in the body provides a continuous antigenic stimulus that acts both as priming and booster doses. When the same animal is subsequently exposed to the same antigen, a temporary fall in the level of circulating antibody occurs due to the combination of antigen with pre-existing antibody. This has been called the negative phase. It is followed by an increase in antibody level exceeding the initial level. This effects of repeated antigenic stimulus forms the basis of secondary humoral response. Now let's know about fate of antigen in tissues. Antigens introduced intravenously are rapidly localized in the spleen, liver, bone marrow, kidneys and lungs. About 70 to 80% of these antigens are broken down in reticuloendothelial cells and excreted in the urine. On the other hand, antigens introduced subcutaneously are mainly localized in the draining lymph nodes, only small amount being found in the spleen. Antigens are presented to immunocompetent cells in two ways, by macrophages and by the dendritic cells in the lymph node follicles. With many antigens, processing by macrophages appears to be a necessary prerequisite for antibody formation. Both macrophages and dendritic cells present the antigen native or processed at the cell surface. Macrophages modulate the dose of antigen presented to lymphocytes so that it may be optimum to induce immune response. Now let's know about production of antibodies. Antigens are presented to immunocompetent cells by antigen presenting cells, in short APC, macrophages and dendritic cells. With many antigens that like T cell dependent antigens such as proteins and erythrocytes, processing by macrophages is essential prerequisite for antibody production, and in case of T cell independent antigens such as polysaccharides, antibody production does not require T cell participation. Only when the processed antigen is presented on the surface of APC in association with MHC molecule to the T cell carrying antigen recognition receptors, that is TCRS, is the T cell able to recognize it. In the case of helper T cells, the antigen has to be presented with MHC cell class 2, and for cytotoxic T cells with MHC class 1 molecules, and the TH cell requires two signals for activation. The first is a combination of TCR with MHC class 2 complex antigen. The second is interleukin 1 produced by APC. The activated TH cell produces interleukin 2 and other cytokines required for B cell stimulation. These include IL-4, IL-5, IL means interleukin 5, interleukin 6, which act as beta cell growth factor, in short BCGF, and the B cell differentiation factor, BCDF, for activation of B cells which have combined with their respective antigen. These B cells undergo clonal proliferation and differentiate into plasma cells that synthesize and secrete antibodies. B cells carry surface receptors which consist of IgM or other immunoglobulin classes. Depending upon these receptors, a plasma cell secretes an antibody class, and that is IgM, IgG or any other single class. However, in primary humoral response, plasma cells initially secrete IgM and later switching over to form IgG. Following antigenic stimulus, not all B lymphocytes get converted into plasma cells. A small proportion of them develop into memory cells which have a long lifespan and can recognize the same antigen on subsequent exposure. The increased antibody response during secondary antigenic stimulus is due to the memory cells induced by the primary contact with the antigen. Antibody production by beta lymphocytes is regulated by T cells. Helper T cells stimulate and suppressor T cells inhibit antibody production. Optimum antibody response depends on the balanced activity of these TS and TH cells. Now let's know about theories of antibody formation. There are two broad groups. One is instructive theories, second is selective theories. The instructive theories postulate that an immunocompetent cells is capable of synthesizing antibodies of all specificity. The antigen instructs ICC to produce the complementary antibody. Selective theories, on the contrary, postulate that ICCs have only a restricted immunological range. The antigen selects the appropriate ICC to synthesize an antibody. Now let's know about instructive theories. First is direct template theories. According to these theories, the antigen or the antigenic determinant enters the antibody forming cell and serves as a template against which antibodies are synthesized so that they have combining sites complementary to the antigenic determinant. Now let's know about indirect template theory. This was proposed by Bonnet and Fenner in 1949. According to this theory, the antigenic determinant enters into the ICC so that a genocopy of the antigenic determinant is incorporated in its genome and transmitted to the progeny cells. Now let's know about selective theories. Side chain theory. According to side chain theory, ICC have surface receptors capable of reacting with antigens which have complementary side chains, which, when foreign antigens are introduced into the body, they combine with those cell receptors which have a complementary fit. This inactivates the receptors. As the compensatory mechanism, there is an overproduction of the same type of receptors which circulate as antibodies. Now let's know about natural selection theory. This theory was proposed by Jarny in 1955. This postulates that about 1 million globulin molecules are formed in embryonic life, which covered the full range of antigens. When an antigen is introduced, it combines selectively with the globulin molecule that has the nearest complementary fit. The globulin with the combined antigen stimulates antibody forming cells to produce same kind of antibody. Now let's know about clonal selection theory. This theory was proposed by Bonnet in 1957. The theory states that during fetal development, a large number of clones of immunological competent cells insert ICCs bearing specific antibody patterns are produced by a process of somatic mutation of ICCs against all possible antigens. Clones of cells with immunological reactivity with self antigens are eliminated during embryonic life. Such clones are known as forbidden clones. Persistence of forbidden clones of their development in later life by somatic mutation leads to autoimmune processes. Each ICC is capable of reacting with one antigen or a small number of antigens. Contact with specific antigen leads to cellular proliferation to form clones synthesizing the antibody. The clonal selection theory is widely accepted nowadays. Now let's know about monoclonal antibodies. First of all, let's know about the principle. Antibodies that are usually produced in response to a single antigen are heterogeneous as they are synthesized by several different clones of cells, that is polyclonal. A single antibody forming cell or clone produces antibodies directed against a single antigen or antigenic determinant only, and such antibodies are called monoclonal antibodies. In multiple myeloma, antibodies are produced by a single clone of plasma cells against a single antigenic determinant, and hence the antibodies are monoclonal. The method for production of monoclonal antibodies against any desired antigen was first described by Kohler and Milstein in 1975. They were awarded Nobel Prize for Medicine in 1984. Now let's know about the technique. Antibody forming spleen cells are fused with myeloma cells to produce hybrid cells, that means hybridomas. The resultant hybridoma retains the antibody producing capacity of the spleen cells and ability of myeloma cells to multiply indefinitely. Animal, usually mouse, is immunized with the desired antigen and lymphocytes are harvested from the spleen. Spleen cells, lymphocytes are then fused with mouse myeloma cells grown in culture which are deficient in the enzyme hypoxanthine phosphoribosyl transferase, in short HPRT. Fusion is done by incubating these cells in the presence of polyethylene glycol, in short PEG. The fused cells are grown in basal culture medium containing hypoxanthine, aminopterin and thymidine. In short, it is HAT medium. Only hybrid cells have properties of both the splenic lymphocytes, in short HPRT plus, and myeloma cells, that is HPRT minus, can grow in the culture. The enzyme HPRT is necessary for nucleic acid synthesis and is provided by the splenic lymphocytes in hybrid cells. Splenic lymphocytes alone cannot replicate indefinitely, while in the unfused myeloma cells are killed by aminopterin in HAT medium. Clones that secrete the desired antibody are selected for continuous cultivation. These hybridomas can be maintained indefinitely and will continue to form monoclonal antibodies. They can also be grown as tumors in the peritoneal cavity of mice by intraperitoneal inoculation, and monoclonal antibodies are obtained by harvesting the ascitic fluid produced. Hybridomas may be frozen for prolonged storage. Mouse monoclonal antibodies, however, proved unsuitable for human therapeutic use because of strong anti-mouse immune response. Human monoclonal antibodies have been developed by modification of the original technique. Genes for particular antibody fragments have been fused to bacteriophage genes. Large quantities of the desired antibody can be obtained by infecting bacteria with the appropriate bacteriophage. Such antibodies hold great promise for immunotherapy. Now let's know about applications. Diagnostic use. Many commercial diagnostic systems use monoclonal antibodies for identification of bacterial, viral and other antigens. Direct fluorescence and enzyme-linked assays utilize monoclonal antibody conjugates. Now let's know about pure antibody. Large amount of pure antibody of defined class can be prepared. Now let's know about factors influencing antibodies production. First is age. Embryo is immunologically immature. Production of antibodies starts after the development and differentiation of lymphoid organs. During embryonic life, clones of cells that have specificity towards self-antigens are eliminated. Immunocompetence is not complete at birth. However, full competence is acquired only by the age of 5 to 7 years for IgG and 10 to 15 years for IgA. Now, let's know about genetic factors. The immune response in different individuals to same antigen varies due to genetic factors. Persons capable of responding to a particular antigen are called responder, and those who do not respond are termed non-responder. The IR means immune response genes control this property. Now let's know about nutritional status. Protein calorie malnutrition suppresses both humoral and cellular immunity. Next point is route of administration. There is better immune response following parenteral administration of antigen than through oral or nasal routes. Now let's know about dose of antigen. An antigen is immunogenic only above a minimum critical dose. Very small dose does not stimulate immune system to produce antibodies, while very large dose may inhibit the antibody producing system and paralyze it. This phenomenon is known as immunological paralysis. Now let's know about multiple antigens. When two or more antigens are administered simultaneously, the effects may vary. Antibodies may produce against all the antigens, or antibody response to one or the other of the antigens may be enhanced, as in triple vaccine, that is diphtheria, pertussis, tetanus, or the response to one or more of them may be diminished. That means antigenic competition. For optimal effect, the nature and relative proportion of the different antigens should be carefully adjusted. Now let's know about adjuvants. Any substance that enhances the immunogenicity of an antigen is called adjuvant. A number of substances such as aluminum hydroxide or phosphate and incorporation of protein. So again repeating, adjuvants, any substance that enhances the immunogenicity of an antigen is called adjuvant. A number of substances such as aluminum hydroxide or phosphate and incorporation of protein antigens in the water phase of the water in oil emulsion delay the release of antigen and prolong the antigenic stimulus. Now let's know about the types of adjuvants. Depot. First is depot. Aluminum hydroxide or phosphate, alum and freons. Incomplete adjuvant, water in arches oil. Second is bacterial. That means freons. Complete adjuvant is the freons. Incomplete adjuvant along with the suspension of killed tubercle bacilli. Chemical. Third point is chemical. Bentonite, calcium alginate and silica particles. Now let's know about the action of adjuvants. Sustained release of antigen from depot stimulate lymphocytes non-specifically and activate macrophages, stimulate CMI. Freons complete adjuvant develops delayed hypersensitivity in addition to enhanced antibody formation. This adjuvant cannot be used in humans because it produces local granuloma. Now let's know about immunosuppressive agents. These agents inhibit the immune response. They are useful in certain situations such as transplantation, when it becomes necessary to prevent graft rejection. Now let's know about immunosuppressive agents including X irradiation, radioemetic drugs, corticosteroids, antimetabolites and anti-lymphocytic serum. Now let's know about X irradiation. It is more cytotoxic to replicate cells and has been used to prolong transplant survival. Now let's know about radiomimetic drugs. They belong to the class of alkylating agents. Example: cyclophosphamide, nitrogen mustard. Cyclophosphamide given for 3 days after the antigen completely suppresses the antibody response. It is much less effective when given before the antigen. It selectively prevents B cell replication. Now let's know about corticosteroids. They cause depletion of lymphocytes from the blood and lymphoid tissues. They are anti-inflammatory drugs that diminish the responsiveness of both B and T cells. They impair maturation of activated cells by suppressing the production of interleukins. However, therapeutic doses have little effect on immune response. Now let's know about antimetabolites. These are substances that interfere with the synthesis of DNA, RNA or both, and thus inhibit cell division and differentiation necessary for humoral and cellular immune responses. They are used clinically in the prevention of graft rejection. Some examples of antimetabolites are folic acid antagonists, analog of purine, cytosine and uracil. Now let's know about anti-lymphocytic serum, in short ALS. Anti-lymphocytic serum is a heterologous antiserum raised against lymphocytes. Antiserum prepared against the thymus cells is called antithymocyte serum, in short ATS. The corresponding globulin preparations are called anti-lymphocyte globulin or anti-thymocyte globulin. ALS is raised in horse. It is devoid of undesired effects and its action is only on circulating lymphocytes and not on lymphocytes in lymphoid organs. It is mainly used to prevent graft rejection. Anti-lymphocytic serum is effective primarily against T lymphocytes. Humoral response to thymus-dependent antigens is unaffected, but the response to thymus-dependent antigens may be inhibited. As ALS is a foreign protein, it may lead to hypersensitivity. Next is we will discuss next video. Okay, thank you.